Great. So as I mentioned, my name is Pyro. I'm the CEO of Smart MCs, and we're here with another episode of our scientific webinars that happens usually once a month. Today we have a very special guest, biomaterial scientist, Kristopher Kilian from UNSW. He is one of our collaborators as well, and has done a really range of great scientific works across the biomaterial science and their applications in many different fields, which he can tell you about all the exciting stuff. But you expect to hear lots of cool scientific kind of talk and concepts, I think, in the courses. And I always have enjoyed listening to Chris's talks. Before I just pass on, so pretty much after this session, Chris will be we'll have like a Q&A session. So if you had any questions, feel free to drop it in the chat, or you can raise your hands. I can let you open your mic. But essentially, the questions will be technical, but also we'll have a little bit of questions on the carrier side of the story, obviously tapping on his wealth of experience that Chris has had on different side of the academic and entrepreneurial journeys that he had. Okay, so maybe I should stop talking now and pass it on to Chris. But yeah, so feel free to drop your questions in the chat.
Well, thank you very much, PR. It's really a pleasure to be here today to tell you about the work that we're doing. So I'm Chris Kilian. You gave me a very nice introduction, so I don't have to go on too much, but I'm in the School of Material Science and Engineering and the School of Chemistry at UNSW Sydney. And I'm going to tell you about some of the work we've been doing. There we go. All right, I think I'll start this story by, really the thing that gets me up in the morning, the motivational context. And so, I'm really a physical chemist by training, but then worked in biotech for a number of years. And that got me really enthusiastic about the area of interfacing materials with biological systems. And when you look at how nature assembles tissue, it's absolutely extraordinary how this complex form arises from these interactions between cells and materials. And so often I think we focus more on the cell side of things, but the materials play a pivotal role because what they do is they allow for the cells to adhere, for them to exert forces and to organize. And these cues actually catalyze a whole range of activities that ultimately lead to the functional outcomes of tissues and ultimately organs and entire organisms. And so, when we try to recreate this, we're somewhat limited. But I think, for me and for many of my colleagues and probably many in the room, we get inspired by this beautiful form-function relationship. And so, we try to use the tricks of the trade, and we use engineering principles and advanced materials to try to emulate some of these things that we see in vivo in living organisms. And so, here's an example from some years ago, a decade ago, by Josh Grohman and colleagues in the lab, where they demonstrated that utilizing a extruded fluid, and if you used a cross-linking chemistry, you could make these meandering filaments. And by changing the flow rate, you could form complex structures. And so using these principles, we were able to form these tubular-like networks that in some ways mimic some of the architectures that we see in vivo. But it never quite gets to the point that nature's had these, millions of years to evolve into these complex structures. So we can get a nice cover image on a journal, but being able to recreate this complexity, we're still a ways away, but we're getting new tricks all the time. I'll tell you a bit about my journey in this way. Okay, so the director of the Laboratory for Advanced Biomaterials and Matrix Engineering, Lab&Me for short. And the way that we try to emulate some of this complex structure is by, as we often have to do, deconstructing into several themes. And so at the heart of everything, we're really wanting to develop new materials, new assays, new discoveries for biotech and medicine. But we have projects that really span bio-inspired hydrogels, which I'll focus on nearly exclusively today. But this is a starting point for many of these things, but we're also interested in ultimately creating microphysiological systems. And so little chip-based devices that can emulate various aspects of physiology or pathology. We have a deep interest in nano and microstructured materials because all of the materials in biology tend to have some hierarchical structure starting from the building blocks. And then we got projects in 3D biofabrication, which is just an outstanding tool for recreating more complex architectures. And then, which I'll touch on a bit today, is using these materials and these approaches to try to build better systems to produce therapeutically useful cells. And so, this often starts with novel polymeric biomaterials. Nature's got a whole complex assortment of biomaterials, and we use plenty of those. But in many ways, as biomaterials chemists, we want to try to deconstruct nature's complexity into simpler building blocks. And the reason we want to do that is because by simplifying, we can better establish structure-function relationships. We can understand how our materials design leads to various activities in populations of cells, for instance. And then from these, we're always looking for opportunities where we can use some of these new biomaterial systems for things like advanced cell culture for manufacture, but also we're very excited about the future of organoid-based microphysiological systems, and we do a bit of mechanobiology as well. And then finally, I think motivating all of this is, if we can create these advanced systems, then we might be able to impact medicine by developing new devices, new therapeutics. Okay, so I'm not going to talk about each of these, of course, today, but I'll give a couple snapshots. With respect to the cell-based product manufacture, we've done some work over the years in utilizing advanced hydrogels for tissue culture. And this is work we did with Sonata Therapeutics, a Melbourne-based company, where we showed that by using complex hydrogels that are coated with various combinations of matrix proteins, that we could essentially prime a therapeutically useful phenotype in the adherent cells. And in this case, we showed that this 10 kilopascal stiffness hydrogel, which is about muscle tissue stiffness, that's conjugated with collagen and fibronectin, we could maximize the angiogenic output from these therapeutic cells. And this work really suggested that we might be able to tune cells to have a wide range of activities just by changing the culture substrate that they're sitting on. Another example is in induced pluripotent stem cells, where we used a soft lithography approach where we micropatterned aggregates of cells on these different gels, now going from about 1 kilopascal, very, very squishy and soft, up to 100 kilopascals, fairly rigid, and showed that by confining cells into these well-defined aggregates, we could coax them to undergo a physiologically relevant transformation. And so they began to undergo development. And in so doing, we could, after about 2 days, you could just shake your flask and the cells would detach from the surface and you get a bunch of uniformly sized induced pluripotent stem cell aggregates. And one thing that we're doing in the lab currently is trying to coax these to differentiate further. And I'll show some work with that using some advanced 3D materials. Another area is our 3D biofabrication. And so Tom Molly started our work in suspension culture. And so this is using microgels or microcarriers that are essentially jammed into a packed conformation where it essentially serves as a yield stress fluid. And in so doing, we could do some direct writing. And so using a 3D printer, we could put in sacrificial inks. And as shown in this little video here, you can see we could perfuse these channels, in this case, just with food coloring, so you could visualize, but we put in endothelialized channels in there to try to mimic blood vessels. A little bit later, Sarah Romanazzo showed that using the suspension technique, we could print ceramic-like materials. And so this was our ceramic bioprinting example. And so because of this jam suspension, you're not limited by the viscosity in your printing. You could deposit these ceramic-based materials in the presence of living cells. And so while you're printing in there, you can see these are cells. This is phalloidin stains for filamentous actin, and blue is the nuclei. You see how the cells just love these suspensions. And these are now gelatin methacrylate particles. And so made with GELMA, which and a lot of the biomaterials people in the audience I'm sure are familiar with, we're able to print within there and then lock it in place. And this seemed to be a really nice mix of ceramics and patient-derived adipose stromal cells that could be used for in vivo healing of bone. We've also worked with Inventia Life Sciences, and this is with Justin Gooding in the School of Chemistry at UNSW, using the Rastrum bioprinter to make high-throughput assays. And we're not going to talk too much about that today, but bioprinters that have different modalities. And so in the first example is using direct write versus the Inventia printer, which is a drop-on-demand, the inkjet-like printing modality. You could have many different styles of printing that could accomplish different feats. And so for high throughput, these are very useful for making, for instance, geometrically structured tissue templates to do high throughput biology. In another project in microphysiological engineering, Tom Molly developed this following on from his bioprinting work. He made these little microcapsules where you could encapsulate enzymes within these microscale volumes. And now these are about one on the order of 100 microns in diameter. And Tom showed that doing this, you could then control oxygen levels in culture. And probably my favorite demonstration of this in our PNAS paper was that if you position these capsules within a single well of a tissue culture plate, you could get a gradient of oxygen from less than 1% up to 15% within a single well. And so really one of the first platforms demonstrating that you could have a gradient of oxygen within tissue culture. And this will be really useful for dialing in the oxygen levels you want within a plate, but also for devices like microfluidic chips. You could imagine you could recreate some of the oxygen gradients that exist in vivo. And then Giulia Silvani, a very talented postdoc in my lab, has pioneered some really exciting microfluidic techniques. And this is where we want to bring everything together, where we make these advanced hydrogels with these clever biomaterials. And then if we could put them in a chip to emulate a complex process like cancer metastasis, then this could be something that would lead to some very exciting biological breakthroughs, but also potentially be useful for drug development. Okay, so that was broad, I know. I just wanted to give a snapshot of the different areas in the group based on this diagram. But the thing that I'm going to focus on predominantly today is some of our mimics of the extracellular matrix. And why are we interested in this? A lot of the natural materials that you can purchase, collagen-based, other biopolymer-based, work just fine. Well, some of the disadvantages of these is that they're so complex, we can't really understand how the physical or chemical properties influence cells. Because they're animal derived, they can be immunogenic, and so they might not work for certain applications like in tissue engineering. And they're really not ideal for many of the things that we want to do, like biofab or cell production, where you don't want to have animal derived materials in your culture. So synthetics are advantageous for a number of reasons. You could control the properties. We understand how they're made, how they're built, how they assemble, at least if we're clever and we use the right battery of techniques. And these are, of course, more amenable to manufacture with these synthetic materials. Some of the disadvantages that have emerged over the years in synthetic realms is that they're often very simplistic. And so, nature uses hundreds of complex proteins assembled in diverse ways in order to reach a final starting point in tissue. That complexity is very difficult to reconstruct, and with our simple building blocks, we sometimes don't achieve it to the degree that we'd like. And also, sometimes you don't get the same sort of properties that you want. But I'll tell you about how we can overcome some of these challenges with some of the work that we've done. And I'll really kind of break this bit down into two pieces. One, our work in 3D, 4D cell and tissue culture, and then extending from that, how we're hoping to use these materials for tissue engineering, cell manufacture, regenerative medicine, etc. Okay, so this story begins with really some beautiful work by Ashley Nguyen when she did her PhD with me. Ashley's now at University of California, San Diego, doing a postdoc in the Aguado lab. And Ashley was exploring via computational means at home during COVID. Various self-assembly motifs. And so I'd asked Ash, could we find some short peptides that assemble in robust ways so that we could use them to coax the assembly of various synthetic fragments? And she discovered this tryptophan zipper motif that has these 4 tryptophan residues that in silico showed that they might be able to form nanofibers. And so she did some mutation analysis based on the earlier published sequence. And this hadn't been demonstrated to be a hydrogelator before, but some of the computer evidence suggested that if you make a couple amino acid substitutions, in particular this valine residue here, that it would show a much greater propensity to assemble. And because we're an experimental lab, of course, it was great because Ash just went into the lab and synthesized this peptide, and sure enough, it forms a really nice hydrogel. And so this was exciting, and so we explored it further. We did some transmission electron microscopy, and we find that it forms these nanofibers pretty quickly, within seconds. These nanofibers then grow, and within an hour, they form this complex three-dimensional architecture, which in many ways resembles the porous structure that you see in natural tissue and collagen matrices and things like this. And so you can see here at neutral pH, you get this spongy-like porous network. And interestingly, I mean, this is microscale now, so this scale bar is 200 microns. We believe that there's this hierarchical ordering going on, and we've done some neutron scattering to prove this. And so what happens is these Trpzip peptides, these 10 to 12 mers, they assemble into beta sheets, which then beta hairpins rather, that then stack, and they form these particles, these disc-shaped particles, which stack and form these nanofibers, which then get entangled. And so it's this hierarchical structuring that turns out gives it some really unique properties, and I'll tell you a bit about those. First, it's mechanics, and so it's really quite an interesting material. If you change the weight content of this short peptide from pretty low weight fraction, 1% up to 5%, you can tune the modulus from hundreds of pascals all the way up to hundreds of kilopascals. And so that encompasses a wide range of different physiologically, pathologically relevant stiffness, where you see in tissue, you get anywhere from hundreds of pascals up to tens of kilopascals. One of the most intriguing bits is that it has this really low yield point. And in many applications, this is it's not what you're looking for. You want your hydrogel to be really stable, but I'll show you it allows for some unique opportunities. And one of which is self-healing. And so this is some thixotropy work we did, where you can see if you shear the gel, this is using a parallel plate rheometer, you essentially fluidize it, it becomes a liquid again, and then it self-heals and stabilizes. You shear it again, and it self-heals and stabilizes. So it shows this reversible self-healing. And I'll show this quick video of how this low yield point gives it some interesting characteristics. You can see it forms a nice gel on the bottom, but then if you give it a shake, it fluidizes. So it becomes a liquid again. And so, you don't want this to happen for something that's like a load-bearing type implant, but if you're trying to culture organoids in 3 dimensions and you want to retrieve them, being able to give a nice flick of the wrist and get them out for proteomics or whatever other applications you're looking to use them for, it could be quite nice. In addition, when we look at some of the other rheological characteristics, we see that one of the interesting attributes is it shows this stress relaxation characteristic comparable to natural materials like Matrigel. And this is important because for a lot of tissue level processes, you need the matrix to have some of these viscous properties, not just stiffness. And so having this stress relaxation has turned out to be really important. And you can see it's comparable to Matrigel. Well, if you have one of the more commonly used polyethylene glycol-based hydrogels, for instance, it's nearly virtually elastic, where you don't see this stress relaxation. Okay, but do cells like it? Turns out yes. And so if we look at viability, cells grown just on the surface of Trpzip on glass versus the gel, they show comparable viabilities. We compared it to another commonly used peptide-based hydrogel, these FMOC GFF gels. You can see that they do far superior in the Trpzip gel. The cells are extending processes, they're spreading out. And in fact, if we compare it to Matrigel, which is the basement membrane derived commercially available product that's predominantly used in so much organoid culture, cells behave very similarly in the Trpzip gel. And we think this is because there's a bulk positive charge at physiologically pH, which allows sequestration of growth factors and matrix. And we proved that by doing some immunofluorescent staining of things like collagen. And after 1 week, you can see we have evidence of collagen being deposited within the matrix. We're still exploring how this works, but it's pretty exciting. And importantly, if we look at long-term culture, where you see these fibroblast cells, after 1 week, you get comparable spreading and extension. And then after a month with myoblasts, you can see that we get really nice proliferation. Importantly, maintenance of shape. And so the entire structure maintained its shape fidelity after months in culture, which is really exciting. Well, for instance, in Matrigel, they dissolved the matrix and collapsed within themselves, which is often what happens in these natural materials. So this means that it could be useful for tissue engineering. So we wanted to look at this as an organoid culture material, and so we partnered with Shafa Waters, an adult stem cell expert here at UNSW, And Shafa has these participant-derived intestinal organoids. And when we put them in our Trpzip, we were actually quite surprised to see a difference in their morphology. And so when these intestinal organoids are cultured in Matrigel, they take this basal out polarity. And that means the inside of the organoid is like the inside of your gut, and where the cells, the surface of your intestinal lining is. While in the Trpzip gel, they're apical out. And so, the apical surface that you would see on the inside of the gut is actually facing outward. And so this is desirable for many applications if you want to study, for instance, that interface at the apical surface. And so we did some probing of this, and here's some immunofluorescence characterization. The cells are perfectly viable in the Trpzip, and they differentiate and they polarize. But we did this forskolin-induced swelling assay to show that their polarity was in fact reversed. You can see in Matrigel, they swell, while in Trpzip, they shrink when they're treated with this forskolin compound. And then as we add in laminin, and so this is we added laminin protein, you get swelling, you get a reversion. And so laminin protein seems to be really important for this apical in morphology. You can see finally at this high laminin content condition, you get swelling comparable to what you see in Matrigel. And that's this quantified here, where you can see the comparison between the swelling characteristics. And so this suggests that we might be able to tune these materials. And so we initially thought it was great that we could have apical out organoids. But of course, for many cultures, you're going to want to be able to control that polarity, particularly if you want to replace Matrigel in a lot of these assays. And so we've been exploring that a little bit further. One way that we've been trying to control polarity is by adding on distal sequences. And so this is our 10-mer Trpzip peptide. But now, rather than stopping at the end of the peptide, we've added on these adhesion sequences. And so the GRGDS, which is from fibronectin, GFOGER, which is from collagen 1, YIGSR, and IKVAV, which are derived from laminin. And interestingly, when we do shear rheology, what we see is that we get very little difference in the mechanical properties when we add on these adhesive sequences. I was surprised by this because many of these peptide-based biomaterials that had previously been developed showed that if you append any other sequences to the end, you disrupt their packing and their structure. So it's clear that where this adhesive motif is being conjugated is not a crucial region for the assembly mechanism. And so, we see pretty much the same mechanical properties. And when we look at organoid morphology, we see a mixed bag of phenotypes depending on what we've used. And so this here is showing Matrigel where you get exclusively basal out apical in, but then when we add Trpzip, we get a mix of phenotypes across the gel. And if we conjugate, if we use laminin or hyaluronic acid or these various peptides, then we can control to some degree this organoid polarization. And so we're still working to optimize this, but we think we've gotten a handle on how we might be able to control this polarity in our 3D cultures. Okay, so moving on from the intestinal organoids, one area that is really important for cell expansion for ultimately production of therapeutically useful cells is induced pluripotent stem cells. And iPSCs are most often cultured on planar monolayers on glass or plastic that have a thin layer of Matrigel or vitronectin coating. And so we asked whether we could use Trpzip to expand these cells in vitro. And so we used our different combinations where we have Trpzip alone or conjugated with the laminin-derived peptide, the fibronectin, the collagen, or the other laminin one. And you can see after extensive culture, this is 12 days of culture, that we get pretty uniform growth of these embryonic bodies in culture. And so these are starting from very small fragments after passaging, and they've then grown to form these large embryonic bodies. And if we quantitate that, we don't see much difference across the different peptides. And if we look at the pluripotency, and this has actually changed. We've now looked for several months, and they seem to maintain their pluripotency. And so we immunostain these for NANOG and OCT4, which are markers of pluripotency. And so these stem cells are remaining stem cells in culture. So this is exciting because it suggests that this three-dimensional Trpzip culture might be conducive for expansion of induced pluripotent stem cells. And iPSCs are being explored for just a wide range of different therapeutic interventions in tissue engineering. And so I think it's a real important commercial need to develop defined materials so that we can grow these things better. But of course, we didn't want to stop there. And so we wanted to explore whether or not these different peptides might be able to influence the cell's differentiation. And so during this culture, we then switched out our regular expansion media that we use for the induced pluripotent stem cell cultures, and we added in a combination of growth factors that's been shown to influence differentiation. And so first example here is ectoderm culture conditions. And so this is going towards lineages of the brain, things like brain organoids or neurons, motor neurons is an area that's really important for regenerative therapies. And strikingly, we see that this IKVAV condition was the best for ectoderm differentiation, with the one coming in second being this YIGSR. This is interesting because both IKVAV and YIGSR are from laminin, and they've been shown previously in other biomaterials contexts to help the growth of neuronal cultures, for instance. And so I was pleasantly surprised to see some congruence with previous work. Then when we look at mesoderm, and so we exposed another culture alongside these two mesoderm induction cues, we saw another hit for catalyzing mesoderm specification. And this is now T brachyury and collagen 4 expression. And we see that the Trpzip GRGDS is best for that. And then finally, for endoderm, the Trpzip alone, so without having any adhesive motifs on the end, seemed to be the best sequence for culture. And so I think this is really nice because it suggests that you can take this scaffolding, the Trpzip peptide, and then you can decorate it with other biochemical cues through conjugation that can help steer lineages towards different germ layers. And so we're now going to try to go further and see if we can use, for instance, our ectoderm conditions to expand motor neurons and our endoderm conditions for intestinal organoids, mesoderm ones for cardiac lineages, for instance. But more work to be done there. We're also exploring the mechanism, and the jury's still out, but we believe it's subtle differences in the packing and engagement of very specific integrin receptors that is catalyzing this predisposition for germ layers. Okay, so that was really an overview of this new material that we're really excited about, Trpzip. And now I'll talk about some of the other directions that we've been exploring for tissue engineering applications. And a lot of the motivation here is based on some of these exciting viscoelastic properties that I mentioned. And so because it has this low yield point, as I showed you, this gives it the ability to be broken up easily through shaking, but also to be extruded by a syringe. And so we explored injectability. And so if you load a syringe with stem cells, and these are now adipose-derived stem cells, into a syringe and you inject them, you see you maintain high viability after injection. And if you look at how they grow, they are perfectly happy once they've been deposited on glass or a gel from the syringe. We also capitalize on this to use this as a suspension printing bath. And so I showed you the granular suspensions in my first slide with the bioprinting. The reality is Trpzip is this hierarchically ordered structure. So they're microscale aggregates that are linked together by nanofibers. And so we use this to do some printing demonstrations, and we showed that you can take a syringe loaded with really any material. Here it's just a high density of cells and media. And you can translate the tip, and because it's a yield stress fluid, it'll fluidize when you're shearing it with the tip, but then it self-heals afterwards. And so we're able to deposit little lines of high dense cell inks and droplets as shown. And so we think it could serve as a really nice medium for bioprinting as well to get some more complex architectures within the material. And then, one of the other really, I don't want to say serendipitous, but exciting attributes that we weren't expecting is its antimicrobial. And so Ash had a hunch based on the tryptophan residues that it might be, and we collaborated with Naresh Kumar's group and looked at its bactericidal properties, and it seems to be resistant to both gram-positive and gram-negative bacteria. And so this means this could be a really nice material for medicine, right? Because it has all these interesting bioactive tendencies, but then at the same time, it's antimicrobial. And so this could be really advantageous as a one-pot material for these sorts of regenerative therapies. All right. Now, in order to use it for a lot of tissue engineering, we need to expand its capabilities. And so one area the group's been exploring is seeing if we can expand its mechanical properties by mixing it with other systems. Trpzip alone forms a rather lumpy gel that is a little bit cloudy in appearance, and so its optical clarity is not ideal, and it's somewhat difficult to work with for some applications. For instance, we'd like to use it as a bandage, but because it's so weak that any jostling or any agitation is going to break it up. And so Yuzi Liu, a grad student in the group, did a really nice job of exploring its mix with polyethylene glycol networks. You can see when you add in the Trpzip, you get more of a honeycomb-like structure. And of course, when you mix it with this rather elastic polyethylene glycol based material and you polymerize it, you see you get much higher stiffness. And so now we go from hundreds of pascals up to tens of thousands of pascals when we tune the amount of PEG. Nevertheless, Yuzi found that it's still self-healing through this very simple test where she chopped a disc in half and then put it back together. If you look at some of the mechanics, we looked at some stress strain, and what we found is that these materials are considerably tougher than the pure PEG, where the pure PEG is quite brittle and it will fracture quite readily. Adding in just a small amount of Trpzip, 0.5 weight%, just expands its mechanics. And then excitingly, you still get some stress relaxation. And so if you look at PEG alone, it's pretty much elastic. But then as you add in Trpzip, you get some more of this viscous characteristics that are important for biology. And when you put cells on these, and so these are the adipose-derived stromal cells again, you see that cells don't adhere to polyethylene glycol gels at all. They remain really rounded up. While when they're put on these Trpzip composite gels, you can see they get very spread and they extend out processes, which is quite exciting because it suggests that this material really does add some bioactivity to this otherwise bioinert material. In another example, Radesh Doshi in the group has been exploring mixing these with various polysaccharides. And the motivation here was the recognition that Trpzip at pH 7.4 has a bulk positive charge. And so we reasoned that if you added in a negatively charged polysaccharide, like sodium alginate, gelum gum, we've been looking at other ones like fibrinogen to make fibrin gels, silk, a range of different materials. And the idea is that you would then be able to facilitate gelation through electrostatics between these materials. And so this works. And so without using calcium, for those of you who don't know, with alginate, which is a biopolymer derived from seaweed, it's a very, very viscous liquid. And if you add calcium ions, those calcium ions cross-link the gel and make it a firm hydrogel. And so we did not use a calcium trigger here. We just took sodium alginate and dissolved it in water and mixed it with the Trpzip alone. And you can see both using alginate as well as gel and gum, you get formation of a gel rather rapidly just by mixing them together at different contents. And so Radesh and the group did some mechanical characterization. And so in addition to being able to mix them together, we saw an improvement in some of these mechanical properties. And so here you can see the blue line and the green line. This is the 0.5 and the 1 weight% Trpzip alone, the storage modulus. When you gel them with alginate, you can see they gel much faster. And so within an hour, you get complete gelation and plateauing. And so faster kinetics of gelation, as well as higher stiffness. See, it's a stiffer material. To provide evidence that it was an electrostatic mediated assembly, we looked at zeta potential. And indeed, you see we got a positive charge in the Trpzip materials alone, negative charge in the gel and gum or the alginate. And when you mix them together, you can see that you can neutralize this charge. The yield point characteristics change as well. You see an increase in the yielding, not dramatic, but still considerable when you're talking about handleability. They're much more easy to handle when they're made with these polysaccharides. And importantly, we see very comparable stress relaxation. And so we're maintaining some of these important viscoelastic characteristics. So one of the things that we're really excited about using the Trpzip gels for, as I mentioned, was cell expansion as well as cell delivery. And so we explored this concept culturing some induced pluripotent stem cell derived MSCs now. And so this is a pretty promising source of mesenchymal stromal cells where you have these allogenic iPSCs and you can differentiate them to this mesenchymal precursor population. And so we cultured them in our Trpzip alone as well as our Trpzip alginate combos. And we see really good viability across the board. One difference we did note that's not quantified here, but I'll point out, is that when the cells are cultured in the Trpzip alginate, they tend to want to adhere to one another more. And so we get formation of these big clusters of cells, as you can see here. And so I think the alginate being bioinert is preventing to some degree the cell's ability to adhere to the nanofibers and spread out and adopt the morphology that we'd seen in previous work. This could be advantageous for some applications. So we'll see. I think we could modify the alginate to make them more bioactive if we wanted to. Radesh is also very interested in delivery of these things, and he wanted to look at extracellular vesicles released from the iPS MSCs. And so he quantified the fraction of EVs that are released into the media from the Trpzip material. And the idea here is that if we can take these Trpzip alginate blends, you might be able to use them as a bandage for wound healing where they would release extracellular vesicles that would be therapeutic. And what he found is that Trpzip alone, you get the sustained release. They keep releasing over the period of a week. And if you add in the alginate, you see that you can increase the kinetics of that release. And so what's happening here, we think, is because of the positive charge, we're able to retain the negatively charged extracellular vesicles that have been secreted from the cells within the matrix. And by neutralizing some of that charge with the alginate, we're able to control that release kinetics. So this might prove useful for wanting to have some type of sustained release for a bandage application like treatment for diabetic foot ulcers, for instance, but plenty of other potential applications as well. Okay, and so that's really all that I wanted to show you today. I hope you found this interesting. This Trpzip hydrogel is one of many potential biomaterials. I think one of the key messages I'd like to convey is that there's so much potential room for discovery in this space. A lot of scientists go to the clinically approved materials or the tried and tested ones like the GELMAs and the PEGs. But finding some of these new novel materials could really open up a broad range of opportunities. And with this tryptophan zipper motif, we've been able to tune the nanoscale and microscale architecture. As I indicated, being able to sequester matrix is a really nice way to get cells to behave in the way you might want. With these tunable yield stress features, we think these materials would be useful for syringe injection, for 3D biofabrication, as a means as just a general matrix for 3D cell culture. With self-healing, I think there's a whole scope of other types of applications. We think it's very promising for long-term culture as well. And then having some antimicrobial tendencies, as well as be able to syringe inject it, I think really expands the versatility. And with that, I mentioned the people that did the work throughout. I just want to thank my group and collaborators, and then funding here. And last of all, thanks for your attention. I'd be happy to answer any questions. Should I stop sharing, Payar? I can't hear you.
Actually, yeah, funny. I was just speaking to myself, I realized. No, thank you for that great presentation. It was really good. I think you can keep maybe the screen on in case anybody had questions, including myself. Yeah, so I think that was really cool to see how spread and different aspects of biomaterials you're kind of attempting to address in terms of the needs. I was gonna start actually with one kind of a high level question first. On you've been obviously attending so many conferences and kind of up to date with all the recent developments in the biomaterials. I wanted to first know, and with an eye for like a new researcher, what do you think are the most hot topics right now? What are the pressing needs right now that researchers are trying to resolve right now? I remember like 20 years ago, there was a lot of stuff happening, or 10 years ago, like with JELMA nonstop, like so many publications coming up. But what are the kind of the state of the art kind of questions now that is always happening?
Yeah, that is a broad question, Payar. There's there's so much. I think, these things evolve, but, some of these questions have persisted, for a long time. I think overall, there's a real pressing need to find synthetic alternatives to natural matrices. There's been this fascinating global shift in the last couple of years where, regulatory bodies like the FDA and the TGA have said, we can't use predominantly these animal models for research exclusively anymore. We need to have these new approach methodologies, these NAMs, to either, support the animal data or even to hopefully replace them. And so I think there's been a resurgence in interest in materials like the Trpzip, ones that, could be well-defined, more amenable to, good manufacturing practice, but support the same level of bioactivity and the same diversity of applications as the natural materials. And so there's a big push for that right now. But, everybody's got their favorite. There's the peptide-based biomaterials group, and then, there's the more conventional long chain synthetic polymers that still show a lot of viability if they're engineered correctly and if they have the right chemistry. And so that's one bit, and one that I'm very excited about, this idea of the synthetic alternatives. But of course, if you go into tissue engineering, and the biofabrication space, I think just every day there's a new publication of an exciting new bioprinting modality that allows the assembly of cells and materials in well-defined ways. That's another area I think that's really important, being able to robustly control both. And I think I started my talk with that, with robustly control both the materials and the cells. Because I sort of feel like, the cell biologists have done a spectacular job understanding, what's going on inside a cell. And the biofabrication community has done a spectacular job of integrating advanced robotics and imaging with some of these technologies. But bringing those communities together is happening now, and it's really exciting. It's really exciting to see, really high level frontier cell biology being done in these complex biofabricated scaffolds. Anyway, I could talk all day about areas.
No, that's great. So a follow-on question to that, kind of, have you explored the world of kind of regenerative medicine and the use of because, like, especially with neo hydrogels, I think there were like a wave of different startups working on using them as a kind of wound for wound healing or injecting for regeneration purposes, as soon as, like, they were seeing some level of promising data in terms of cell growth and how cells like an hydrogel. And I guess startups or people, experts, divide into a few different groups. Some believe in cell-based therapy, some believe in the body should kind of heal itself. Have you explored those with Trpzip or the other hydrogels, like the PEG ones that you were showing?
Yeah, we're trying to. I mean, I know what you mean. I mean, there's different camps, I suppose, that think about doing these things differently. I mean, the trouble is that getting something like this through all the regulatory hurdles, once you're implanting a new material, I mean, it's a long road, right? And so I think if you're a researcher like myself, or particularly someone who's starting a company with this sort of technology, you got to recognize that it's going to be a considerable investment that you're going to need in order to follow that path. And so oftentimes I think it's, if you've got an idea that is going to help patients faster, and you can use something that's clinically approved, like the polylactic acids, polyglycolic acid-based degradable polymers, then by all means, that's the route you should go. The issue, I think, is that as we get more sophisticated in our means of regeneration, PLGAs aren't going to cut it. And so we're going to need to have more complex materials. A lot of the issues, I think, is that, when we still don't really have a full understanding of what happens to cells when they get put in vivo, right? When you're talking from either an allogenic or an autologous cell source, the trouble being that the in vivo environment is so complicated. And, a lot of the early work at, transplanting mesenchymal stromal cells in cardiac disease, for instance, they saw therapeutic efficacy in one trial, and then the next one they wouldn't. And then the animal trials would be successful, but then the early clinical trials wouldn't. So it got really, really a bad space for a time. And I think at the end of the day, it was understood that, well, you get such a huge degree of cell death after you've injected these cells. And so development of materials that are supportive, that keep the cells viable and allow them to do what you want them to do is still very important unmet need.
Yeah, certainly. So I think, yeah, like probably a different road slightly to the kind of the more non-animal models, like a different route, different type of activities and things needed to before it can be commercialized. But yeah, no, that's very cool. Thanks for that. Let me just unmute Gulam. I think might have a question. Hopefully doing the right thing. Ask you to unmute. Is that working?
Looks like we got one in the chat as well.
Yeah.
Yep. It seems I'm okay.
Yeah.
Yeah.
Nice to hear from you.
Go ahead.
Hi, Chris. First of all, super inspiring talk, but that's not surprising. I've got like many things that came up with what you said, but I'll just focus on one. Like I'll try to be short. Did you check the immunogenicity of your Trpzip?
Yeah, we just recently did. The jury's still out on the full data set, but we don't see any like bad inflammation with the blood studies that we've done. And so we've done some whole blood studies recently. This is unpublished. You know, I'm just sharing because I'm that kind of guy. But it seems that there is an inflammatory response, but it's not one that we're too concerned about. I'd have to ask Radesh. He did those experiments just within the last month. But we haven't done in vivo experiments yet. We're planning those right now. We've got some collaborations with a burns unit here in Sydney where we're going to do a pig model, and that's where we want to see where the material goes. We've tracked it in vitro, and it does seem to diffuse over time. And so my fingers are crossed that it's going to be one of those situations where the cells are depositing matrix and it's assembling into some sort of fibrous structure, and then the Trpzip's going away.
Okay.
Our early work with blood suggests that there's no catastrophic immunogenicity.
Okay. Well, promising. Can you still hear me?
Yeah,
I think. Yeah. I mean, if possible, I'll write to you later on because like I really have a lot of things to ask you.
Yeah, let's chat. We don't
with pleasure.
all the time.
Yeah.
Thank you, Gulam, for that. That's great, and love that a long conversation. So that's great. We have another question in the chat. So we've got, so you talk about the sponge-like morphology of the Trpzip hydrogels and show the distribution of the cells on the surface of the gel. Were you able to identify if the cells remained on the surface of the gels, or were they more embedded within the sponge pockets? Will this affect tissue regeneration?
Yeah, no, great question. The cells love it in 3D. They're perfectly happy in the Trpzip. We did that one particular example. The only reason we culture on the surface because we started with a PEG chemistry that required radical initiators that cells don't like. And so we had to form the composite gels first and then seed the cells. And so we did an early two-dimensional characterization. But we've since moved on to more gentle chemistries. And so we're exploring the polyethylene glycols with the maleimides, where we have the dicysteine terminated peptide for the click, quote unquote, and they're perfectly viable and happy there. And they're in 3D in all of the alginate and gel and gum cases that I showed. And so they really like 3D, and it'll certainly affect how they behave in vivo. I mean, we find the cells can move through the material. They don't travel too much from what we've seen. But at the low weight content, the 0.5 weight percent, they certainly spread out and move and migrate around. One thing I'd love to do is try to impose a cytokine gradient and see if we could force migration through the material, but we haven't done that yet. Hope that answered the question.
I'm sure it did. That's great. So also then a kind of a okay, we got a thumbs up, so that's great. So kind of a bit of other now I think we can slowly move on to the career side questions. And I have a lot of questions, but I think we can keep it short. But with an angle of a new, fresh, imagine student or someone who's stepping in this field, what do you think is kind of the best ways of choosing the pathway? So I guess it's really related on the research group as well and the settings that they run. But what is the best way to find a good research question, essentially, in your experience in the past many years?
Yeah, no, that's a good question. I think first, read a lot. So it's important that you understand the literature. I think with AI and the way that everybody's so busy all the time. People have difficulty finding the time to read. And so making sure that you read the literature, because you'll find little nuggets in those papers that aren't clear from summaries from AI or something like that. And so identifying those gaps is a key bit, right? And so you want to read the literature and you'll find out where's there an opportunity. Another bit is just having confidence in creativity. And so looking at a field, and if you have an idea, even if it seems crazy and your friends tell you you're nuts, believe in yourself. Believe that it's a good path to follow, because these crazy ideas sometimes come to fruition and lead to really exciting results. It's one of the best things about an academic career, I find, is being able to pose these sort of questions and finding a career in industry where you have some of that flexibility to spread your creative wings is also good.
No, yeah, no, that's totally I can relate to. So one question, do you think you'll need a PhD to be successful in this field?
I don't think so. I think it depends on what your ultimate goals are, right? I mean, for me, I started working in industry after bachelor's, so I had no interest in a PhD. And so I was working in biotechnology, developing some of the first generation DNA microarrays. And so I won't say when, because I hate sounding that old, but I was synthesizing oligonucleotides on glass slides. And this is the first generation of Agilent's inkjet microarrays. And I loved it. It was so rewarding. And I went down leadership track. And so I supervised a team of 5 people for a short time. But then I started collaborating with some of the senior scientists, and I saw that the way that they were working was quite different. And I recognized that one of my strengths is being able to have some of these creative instincts in a broad perspective. And so I wanted to fan those flames a bit. And so that's when I went back for a master's and ultimately a PhD. But I don't think it's necessary to be fulfilled in this field. In fact, in Australia, we've been talking about this recently in the context of Australian industry. I think that we need more people that are bachelor's, master's level trained to really realize the future of high-tech biotechnology. And this is probably true globally, because the PhD is really great for training you to pose and to answer these complex questions, but it's not absolutely necessary. And so I think for doing things like optimizing procedures for GMP manufacturing, bachelor's, master's level is desirable. And so I think it depends on what ultimately what you want to do, and actually where you're suited as well. We all have to self-reflect periodically and figure out whether it's time to stop or whether we should keep going in our education.
Yeah, yeah, and I think partially follow on to that just before I pass it on to Rebecca for her question. I think there's a lot of times like the time has passed like quite a lot in the past 10, 20 years, and the academic kind of settings has changed as well. I think maybe the competition's got more or maybe less, I don't know. So what has been kind of your observation? Do you think like fresh graduates now get the same opportunity as they would get like 20 years ago through like becoming after a PhD, like doing a postdoc and becoming a lecturer or supervisor, or has it changed a lot?
Yeah, I feel like it has. I feel like unfortunately, I don't want to be negative at all, but we really need to convince the public and the governments that they have to support basic science, because the trouble is that there was a few times where it was really exciting and really promising for fundamental research being carried out, and I just the support isn't aligning with the training, right? And so we have all these fantastic PhD candidate, not PhD candidates, PhD scientists that are looking for jobs, and the opportunities aren't presenting themselves as they did maybe a decade ago, I think. And so but this is a global problem. I think science is not viewed so favorably in many circles, and we have to constantly think about communication and getting the public to understand that what we do has merit, and that these types of careers will lead to the innovation that's ultimately going to make lives better.
So it's kind of, I guess it's been a bit of downhill.
I think in some ways, But what as always happens, these things are kind of go in cycles. And so I think that's been recognized, just like what I mentioned in my last comment with the recognition that we need more bachelor, master's level trained scientists in Australia if we really want to make an impact in things like biomanufacturing. And so, once that's recognized, and as long as the message is carried clearly to, the right people, then hopefully there'll be some shifts where those opportunities will increase. And I think it'll happen. I'm always an optimist with these things. I have seen, I've had talked to a lot of PhD level scientists that have been a bit dissatisfied because of the struggle, because it's challenging to find that opportunity. And you got to be flexible when you get to that level, because you're so specialized that it's very challenging to get that niche positioned. And oftentimes you got to cast a wide net and be willing to move. I mean, I certainly did.
Yeah, and you might move away from what you actually want to do. So yeah, I can hear you that. So pass it on to Rebecca. I'm gonna ask to unmute. We have one more question after that as well. Rebecca.
All good. Hi, I'm Rebecca. This is my first time listening to like this type of talk. I thought it was amazing.
Thanks, Rebecca.
You were such a strong talker. I found it so inspiring.
Oh, I appreciate that. I thought I went too fast because I usually do, but
no, you did great. I am currently a student at UTS, and I thought the conversation you just had with Paya, like that is exactly what the questions I'm asking now as well. But it's good to know that you don't have to do a PhD to enter this type of space, because I would love to. Yeah.
It depends on what your goals are, and it's different training for different jobs. A lot of people think the PhD is all about the things that you learn during the degree, but it's really not. The PhD is all about the way that you think. And so you're put through this really intense program of study where ultimately what we being, people that hire PhD level scientists want is someone who could critically analyze a problem, not get discouraged, maintain, confidence and be creative. And so it's something that, you really need to kind of go through the ringer to hone those skills. But
yes, but it's exciting that like you can still, like, industry is still inviting this like innovation space.
Oh,
yeah, you can do more research. I've just come from an interview with Smart MCs.
Oh yeah.
And I thought I saw that you had the seminar and I was like, oh, I'm gonna see what you guys do. But I'm really interested in this. Anyway, I'll question on to somebody who's got more.
And Rebecca, I think Chris, and Chris always looks for good PhD students, so you can consider that as well.
Yeah. If you decide that's the route, get in touch.
Yeah, fantastic. Thanks, Rebecca. One more question before I let you go, Chris. It's been so engaging. Everyone is loving, and I love the questions as well. So one more question. So following this research gap question, so I'm wondering, out of the millions of directions to go forward with biomaterials and biochemistry, how do you decide which material to mix to try out and which research direction to go forward with? Like, how do you figure out the magic of Trpzip?
Yeah,
I guess a bit of methodology.
Interesting question, and I think when I got started, I really thought it would be more directed. But there's a lot of it that's serendipity. There's a lot of it that's just being in the right place at the right time. People don't mention that as often as as it's a reality, because what I do is I look at the things that I'm excited about, because I think in a lot of these areas you have to have passion for the work that you're doing. And so if you're looking at an area that you're super enthusiastic about and you read it in depth, then you'll find those openings, and often it's just instinct, right? Or you're like, what, for this I was dissatisfied with the materials available for self-assembling systems. And so I was like, let's try to find a unique self-assembling system. But I wasn't expecting a pure nanofiber-based material. That was really because Ash is so clever, and she was doing some of this computational modeling, and she came upon this sequence. I wanted something that I could stick to the end of my PEG gels that would help them have a soft dissociation. And that doesn't work so great with the Trpzip. But Trpzip makes a great nanofiber. So that was a bit of a surprise. And this happens a lot in science, I think. My feeling is, do something that you're excited about, find out, where effort is needed, where, I think having sort of a long-term goal is helpful. I don't think it's absolutely necessary. I think people should still be interested in very simple fundamental things, but if you're interested in delivering cells to somebody who experienced a heart attack, start there and then go back, look at what are the materials that are available, what's their problems, what's missing, what do we need to develop, and from that your list gets narrower and narrower. But often the discoveries aren't what we started. My PhD plans, when I get to sit down with a student right out of the gates, is rarely the structure that it is at the end. Right? We hammer together some ideas to get started, but I mean, science is so dynamic, and sometimes your ideas aren't that great, and you'll find new ones once you're doing the experiments.
Yeah, that's very interesting. And beside the coincidence model, I think one thing that I noticed from your answer was the different skill set. So you mentioned Ash, the student, was actually so good at the computational stuff, so she was probably good there. And then you knew exactly what you're looking for, self-healing kind of mechanism. So I think that's probably one of the key things in science as well, where you can actually, that's where the beauty of collaboration and talking to different experts, I think, really can help.
Absolutely.
And different skill sets can bring in. But look, that's fantastic. I don't want to take more of your time. I know we already over time, but I could continue this forever. I think we'll have a lot of more cool questions, but I guess we can hopefully host you later on as well for more questions. Thank you so much for the time you have put in here. If anyone had any questions, you can shoot Chris an email, and I think everybody really enjoyed the talk, and I'm sure on YouTube they will be able to also send you an email if they had any questions.
Cool. Great. Well, it's been my pleasure. Thanks so much, PR, and thanks everybody for coming.
Great. Thank you, Chris. Have a good rest of the day.
See you later.